All right, so welcome everyone. Got a wonderful talk on orchid fungi with Ellen Garcia. It's very important. Orchids probably wouldn't reproduce by seed without those fungi. So Amir, I'll hand it off to you. You can introduce Ellen, please. Thank you.
Sure. Thank you, Alan, and thank you, Javier, for the invitation. And thanks for everybody who's here today. Ellen joined. Ellen is a graduate from Earth and Environment Department with a Bachelor of Science in Environmental Studies, and she also was in Agroecology certificate program. She joined our graduate program in fall of 2019, and even before that, we were chatting with Jason, and she was working at Fairchild Tropical Botanic Garden in order to come up with a research project. Since then, she's been working very hard, sampling a lot of different orchids from different locations that she will be talking about it. And she also is a recipient of several different travel grants and prestigious grants that she received so far. So she's going to talk about technically her proposal on a study of symbiotic relationship between mycorrhizal fungi and South Florida native orchids in different microclimates. So without any further ado, Ellen, please.
Thank you. Thank you for introduction. And I'm very happy to be sharing Thank you. So the title of the study is the symbiotic relationship in mycorrhizal fungi and South Florida native orchids in different microclimates. And this photo to the right is just of Encyclia tempensis and a photo I took in Naples Botanical Garden. How do I? Oh. So a little introduction. Orchidaceae is one of the largest families of flowering plants with about 25,000 species worldwide, and they're known for ornamental value, medicinal, and cultural values. But however, due to overcollection and habitat loss and human exploitation, they've been severely threatened. Many species have become threatened, and it's estimated that 10% of all orchid species worldwide are threatened. And these are some photos of my research sites. The top photo is at Naples Botanical Garden. These are naturally occurring orchids. And this is to the right of in the top right is the site where they were located. It was somewhat like a pine rockland. And the bottom photos show in Doral where it is, they were planted from the lab from Fairchild Tropical Botanic Garden into downtown Doral Park. And this is just an urban area with streets all around. So more information about mycorrhiza. Due to the lack of endosperm in orchid seeds, they rely strongly on a specific species of mycorrhizal fungi and also rely on unique pollinators. And orchids are also sensitive to environmental factors, especially, and this may limit where their habitat can be. Another important factor is climate change, and things like an elevated CO2, drought, and the warming have been shown in studies to have an effect on plant microorganism interactions. So orchid species have been found to have the obligate relationship. So many studies have shown, specifically with the species Telesnella, to be able to support germination for the seeds to be able to even grow, and then in some cases continue to support into adulthood. And these photos are from at Fairchild Botanical Garden, where I looked at the mycorrhizal fungi under a microscope from collecting the root samples of orchids. So the photo number 1 was the setup. Photo number 2 is the actual mycorrhizal pelotons that are small and like globular structures that are white to cream. And then the third photo is these pelotons after they have been plated on the media, and that's how they grow. And then in the fourth photo is a subculture from the number 3 grown in liquid culture. So orchid breeding programs have focused on planting these orchids back into their native ranges, and this is done into urban areas and like non-natural landscapes and into botanical gardens. These lab propagated outplants are especially prone to mortality, and it is necessary to target these conservation programs for maximum efficiency. So in this case, our native host trees in these urban and botanical garden settings are potential suitable habitat for these sterile lab outplants. And these photos are again from Fairchild, and these are the lab outplants and the seedlings when they're being grown in the lab. And they're grown asymbiotically. They rely on the media to get their nutrients instead of the mycorrhiza. So the questions I have for my research are, what are the best management practice guidelines for planting these orchids from the lab into an urban and botanical garden setting? And second, are the necessary fungi available in these non-natural landscapes to support objectives are to evaluate the correlation between phenotypic traits that are measured and the optimal growing conditions in various microclimates. And second, to investigate fungi communities for orchids that are planted in these urban and botanical garden settings. So the species that I am looking at are Anthurium insatum, Bletia purpurea, so Anthurium insatum is in this lower region, and Encyclia tampensis has been seen in this wider range, and Bletia purpurea in this lower region, as well as sort of Podium punctatum. And I just want to highlight here the Florida state government status for each of the species that I am looking at. Encyclia tampensis, which had like the larger native range, was commercially exploited. Pseudopodium punctatum as endangered, and Bletia purpurea is threatened, and Anthurium insatum is also endangered. So some information about my sampling sites and the host trees that I was looking at. One of the sites was at Fairchild Tropical Botanic Garden, and this was considered a tropical savanna, as shown in this Copenhagen climate zone. It would be this lighter blue section, and Naples Botanical Garden on the West Coast was also a tropical savanna under this climate classification. And downtown Doral Park, as I showed photos earlier, these have lab outplants, and they would be characterized as a tropical monsoon since it is more inland and to the north, so it's in this medium blue area. And the three kinds of host trees that the either naturally occurring or lab outplants were planted on were live oak, cypress trees, and palm trees. And so the data collected when I did the field sampling at those three sites and on the various host trees was measuring the chlorophyll content using non-invasive handheld devices like the SPAD and Green Seeker. And this has been used a lot in agriculture to measure nitrogen and possible deficiencies in their growth. So this can be a measurement of the plant livelihood. Other measurements were orientation, the height on the tree, the light intensity, number of leaves, number of roots, and leaf length and root length. So the question here is, how does the host tree and the environmental factors and mycorrhizal fungi affect the plant health? And this photo is from sampling at Naples Botanical Garden using the SPAD device. So my methods are sampling, as I mentioned, of the of the naturally occurring and lab outplants. And when I sample them to take these phenotypic measurements, I also took about 1-inch root sample and looked at it under the microscope. And this is how it would look like under the microscope, and scraping off the outer layer of the root. And from there, the pelotons disperse into the water, and I can see them under the microscope, and using a pipette, I can culture them and they will grow within a few weeks. And from these cultures, either liquid or from liquid cultures, and from the root DNA as well, I can perform a DNA extraction and a PCR of 3 fungal ITS regions. Some of the other processes are PCR cleanup and a sequencing reaction. And this is done to be able to sequence them at the FIU DNA core facility. And once receiving these sequences, a tree can be designed using the Genius software. And the PCR of the 3 fungal ITS regions is to scan for different types of fungi. One is for the kingdom fungi, and one is for other orchid mycorrhizal fungi. And the third primer is specifically screening for Telesnella to amplify this DNA if it is present. So my preliminary results so far, I was able to analyze about and the results that I obtained was that there was 2 distinct classes found in my results. And this is a mixture of Encyclia tempensis and Certipodium punctatum. And in one class of my data was Ascomycetes, which are suspected pathogens. And there needs to be more research on the functionality of that, but I wanted to highlight that I did find that there was the class of Basidiomycetes, and it was Telesnella, and this was separated into 2 clades, so 2 different clades of Telesnella. And this shows that both at Fairchild Botanical Garden and at Doral, these fungal species that have been seen prior to germinate and allow for survivorship and be functional in the orchids was found in these sites. So this is very promising for breeding programs, This is very promising for breeding programs to know that in sites like at Doral, these native host trees that we have, such as cypress in the bottom and oak trees, are providing beneficial mycorrhizal fungi for the orchids. I also want to highlight that in previous studies done at Fairchild, the Telesnella that I found was seen to be very similar to the one that was previously found. So it was interesting to see also that they were found on the varied host trees and even in the different microclimates. So some of my next steps is that I have to do the statistical analysis on the phenotypic data that I obtained using SPSS, and also finishing processing the rest of my sequencing data for the other sites like Naples and the other species, and using this information, creating a different phylogenetic tree analysis. And this is another photo of an orchid fungal culture. That's it, and thank you. If you have any questions, please let me know.
Hello, may I ask a question?
Yes, please. Happy to.
Nice presentation, Ellen. Did I understand that the very same fungus occurs in 2 different genera of orchids?
So in two different, the two different species of orchid? Is that what-
Yeah, 2 different species.
So for some, yeah, for this that I'm showing here,
Yeah.
for Pseudopodium punctatum and Encyclia tampensis, they both were using a very similar Tillisnella fungi.
Neat. Yeah, that's what I thought. How cool.
And also on different host trees as well.
So the word I used was genera, genus. So for Encyclia is one genus and Pseudopodium another. Very cool. They had such a similar one. Thank you.
To follow up on Suzanne's question, this is Gene Rosenberg. The mycorrhizae are specific, well, they relatively specific to the orchids, right? And well, I mean, I'm just wondering in general, what are the fungi doing on the host plants when they're not in symbiosis with orchids?
That's a good question.
Good question.
Yeah. So they're not in symbiosis with orchids. That's a good question. I have to think about it for a second. And then they would also be with other plants. So in this case, these are epiphytes, but the other two species that I have, they are terrestrial. So they would be interacting in a different kind of network. And I've seen in other research the connection that the mycorrhizal have, and that their abundance also relies on when there can be some there as well. So I'm not sure to what extent do they also rely on the orchids to be able to thrive in a certain location, or if it's based on the host tree itself and the network that it has in that like microhabitat.
You know, I'm just wondering, how the mycorrhizae make a living when they're not,
Yeah.
with their orchids. So, because the orchids pick them up from the,
Yeah.
from the host plants that they're growing on. But so I was just wondering. Thank you.
Yeah, no problem. And there's also, I think, research about just the mycorrhiza not giving back to, the orchid not giving back to the mycorrhiza as well. So the orchid sort of just taking from the mycorrhiza and not giving it any nutrients as well in some cases. So that would be like a more in-depth research on how the mycorrhiza functions in it.
Mm-hmm. Thank you.
Thank you.
Yeah, and Jason could also chime in. This has been working on mycorrhizal for many years now.
Yeah, sure. Yeah, she's pretty much right on. There's still a lot of our understanding what the functionality and the natural behavior of these fungi are in the epiphytic realm. One thing we do know is that these seem to be ubiquitous fungi that are always around. A lot of these, when they produce the fruiting bodies, they're saprophytes, and even sometimes can be decomposers. So, but again, we don't know if they're pathogenic all the time on the tree or if they're just benign mycelium. And no one's really linked the fruiting bodies to the mycelial phase of these fungi as well. So again, the below ground, the terrestrial realm is much, we know a lot more of that, but a lot of research needs to be done on the epiphytic realm and what these fungi are doing when they're not interacting with the orchids. We do pick them up on bark samples though, so they're around, but usually as mycelia.
Thanks, Jason.
Any other questions? All right, I guess that.
Well, I have another question then. What do you grow the fungi on in the, you're growing for the Million Orchid Project, you're growing the orchids out in the laboratory without the mycorrhizae, right?
Yeah.
Mm-hmm. And so what do you grow the orchids on when in that project in the culture? And likewise for the mycorrhizae, what do you grow them on? And in your in the laboratory.
Yeah. So they're just specific nutrients to enable their growth. I don't remember at the top of my head what it has. For the orchids, I'm not the one that is propagating them, but I have made the media before, and it was just a mixture that I guess was readily available for them to be able to absorb. And for the mycorrhizal fungi, it's a media made of like 10 different like micronutrients that would enable their growth. PPE media, but yeah.
Yeah, just to answer the first part of the question that Jean asked, for micro propagation and tissue culture, there are many different medias we can use, macro micronutrients, plus different PGRs, plant growth hormones, different families of auxins, different types of cytokinins or gibberellins based on what we are looking for, it based on the somatic embryogenesis or non-somatic embryogenesis or different plants that we have. We can use different types of the combinations of the PGRs. But what Fairchild is mostly using for seed germination is fixed media, which is macro and micronutrients and a little bit of plant hormones. But again, I mean, there are different functionality for tissue culture. If you want to have roots or shoots, and definitely those combinations of the hormones will be different. It needs to be adjusted for each different species.
Thanks. I mean, I've grown algae in lab cultures, and so I was sort of curious about how that would work with orchids, and on one hand, and with mycorrhizae on the other when they're kept separate. Thanks.
Yeah, just to add on to that a little bit. So yes, we do have a species-specific cocktail for each one of the orchids that we grow, each species of native orchid and other things. And a lot of that research was done by our high school program. That's what they test, is different plant growth factors. So specifically for Encyclia, we had a auxin supplement to a commercial available media. And the black colors from a charcoal, which is an additive that they put in there. And we use that for Encyclia. For the fungal media, she mentioned, this is a specific kind of custom blend for orchid, for this group of orchid mycorrhizae in the Basidiomycetes. And that's a cocktail that we've published before, and it's called BS or PE media. It's essentially glucose, minimal salts, and agar to solidify it. But it does, we don't have things like peptone and different proteins in there to prevent bacteria. So one of the things we do is we want the fungi to grow, but not the bacteria. So we can kind of tweak that blend. But she should be able to present that in the in her defense in the methods, the exact formula.
And why not grow the? I'm just curious. I mean, why not in the laboratory before you outplant them? Why not grow them with the mycorrhizae that they would normally be found with, in the laboratory? So you outplant them with the mycorrhizae.
Great question, Mike. And the main reason is the difference in growth rate. So you can imagine that the fungi grows at an exponentially faster rate than the plant, and essentially will just overtake the little seedlings. And that's been a conundrum with a lot of orchid growers thinking about how to get these things inoculated beforehand. So we've actually done some work with that, where we've taken the sterile orchid and actually inoculated it with a liquid fungus on the, like spraying the roots with the liquid fungus and then planting it into a greenhouse situation. But yeah, that's the main issue in vitro, is that the fungus just grows so much faster, it will just overtake the seedlings and consume all the sugar.
That makes sense. Thank you. I was just curious.
Yeah, that's the main reason. But we're working on that. We're working with things like lean wood. So using just wood in agar, something that the fungi can break down, but it will also kind of slow its growth. So we are working on things like that. And I think that's some future exciting research.
All right. Anyone else? I guess that wraps it up. Thank you, Ellen, for that insightful presentation on what kind of fungi we're finding with these outplanted orchids. Very cool.
Thank you. Thank you so much, everyone, for listening.
Good job, Ellen.
Thank you.
Yeah. Thank you, Ellen. Bye, everyone.
Bye.